So welcome my dear students. So in this video lecture tutorial we are going to discuss about biofilms and quorum sensing. Okay, so quorum sensing and biofilms, these two are very important topic in industrial microbiology and their applications. So now before going to start about the quorum sensing and what are the biofilms, what are the applications and the basic concept, we need to know regarding the socio microbiology. This is a very new term in socio-microbiology, and socio-microbiology is a part of broader discipline of microbiology. Okay, and the study of organisms, bacteria, yeast, mold, viruses, and protists. That is nothing but the microbiology that cannot be observed with the naked eye. Is understood? You cannot see in the naked eye. You need the microscopic instrument or microscope, but having critical mechanisms for propagation. But all the microorganisms are having the critical mechanisms of microorganisms, okay, for propagation. And socio-microbiology means it's a border part of discipline means in our daily life we are exposed to different types of microorganisms like bacteria, yeast, molds, and viruses, and how we are getting infected, how we need to culture of useful microorganisms for industrial application purposes. So these are the things are contains in the socio microbiology chapter. The term actually socio-microbiology was introduced by the scientist Matt Persek and Peter Greenberg in 2005. It's very recent, not more than only 15 years back. Trends in Microbiology is the journal, and here is the reference. They published the term and refers to the group of behavior of microorganisms. So in this paper, in this journal, they have published a paper, Trend in Microbiology is the journal name, international journal. And in this volume, page number, you can find the authors, scientists, Matt Persek and Peter Greenberg in 2005, they have actually refers a group of behavior of microorganisms. Okay, two topics that form the core of sociobiological research and microbial biofilm formation and cell-cell communication. Okay, so all the microbial behavior and their socio-microbiological research including and their biofilm production, all the things are highlighted there. This is the good reference to understand, to better understand the socio-microbiology and quantum sensing. Now, uh, at the introduction part, what is actually mean by quorum sensing? Quorum sensing is cell-to-cell signaling. So, is the quorum sensing is nothing but suppose you are having a mass of bacterial cells. When cell-to-cell signaling is passing through from one cell to another next to the bacterial cell, the collectively controlled by gene expression is nothing but the quorum sensing. Sensing means sense. Suppose some cells are there. Okay, so from one cell to next cell, another cell, signal is passing. Any kind of cellular signal is passing. So signal is related to gene expression. Okay, so these signals is collectively called quorum sensing. This type of bacterial communication, this is the nothing but a communication system, is achieved only at higher cell densities. Means if you have 50 cells, 50 number of cells, you will not able to find the quorum sensing. But if you are having 5 lakh cells, okay, so more than 1 lakh cells, bacterial cells, means the higher cell densities where the quorum sensing data can be recorded. So bacteria release various type of molecules cause as auto-inducers, means and in the extracellular medium, these molecules are mediator of quorum sensing. So mediator, so quorum sensing is nothing but the process by which cell-to-cell communication signal is passes. Okay, so in between two cells, there must have some medium. So medium means this is nothing but the bacterial extracellular medium. Extracellular medium means they actually secrete some inducers. These inducers are actually act as the quorum sensing mediator. When concentration of this signaling molecule exceed a particular threshold value, these molecules are internalized in the cell and activate particular set of genes in the bacterial population. So that means that signaling molecule should not exceed a particular threshold value, because if suppose the extracellular inducer molecules are higher densities, so higher densities means it will be very difficult to pass all the quantum sensing cell signaling or communication process. So if it has a certain limit, so that the cell can pass through the communication message to the next cell. So here you can see the microbiological one plate, that is nothing but the fungal plate. You can see here the study of group behavior of microbes is nothing but microbiology. Socio-microbiology advances the understanding of microbes. What the type of this contain? The study of group behavior in microbes, debate over environmental versus genetic determinants, biofilm and quorum sensing, concept clearing, model for dissecting social behavior at a genetic level. So these are the major areas where you can explain the things. Now you can see that the bacteria are dynamic creatures. So here you can see the different types of bacterial culture. Actually they secreted the different extracellular matrix, extracellular inducers or extracellular compounds. Bacteria are dynamic creatures that are able to regulate their metabolism and lifestyle to respond to a variety of environmental cues. This quiz includes changes in their chemical, physical, and biological surroundings. In recent decades, microbiologists have come to appreciate the bacteria are even able to recognize changes in their own population density. Yes, cell density dependent regulation has been termed quorum sensing. Okay, so you can see the different color. So different color indicate means different metabolites, secondary metabolites or secretion is there. Okay, so microbes run in the body as a normal flora. So that means it is nothing but in our body, microbes run much in our body, because in our digestive tract or in any organ, you can see there are lots of microbes, particularly the useful bacteria. And the human microbiome in our gut, mouth, also you can find bacteria, skin, and elsewhere harbors 3,000 kinds of bacteria in 3 million distinct genes. So just you think, 3,000 kinds of bacteria is actually harbors the human body, the gut, mouth, skin, and elsewhere. Elsewhere means on the skin or elsewhere. Okay, from top to bottom. So our own cells struggling by on only 18,000 genes and so far on, because in the human genome this biotech century will be microbe enhanced and maybe microbe inspired, comforting a difficult problem. We might fruitful ask, what would a microbe? So that means the important thing is that microbes in our body can be hair, gut, mouth, and skin, and more than 3,000 kinds of bacteria is there in our body. Okay, so this is the overall of concept of quorum sensing. So in the next slide, we can able to think about the detailed procedure and detailed signaling pathways and the mechanism. Okay.